Welcome to Executive Insights for Life Sciences Innovators, and thanks for joining us. On this podcast, we'll explore leadership, innovation, and growth in the life sciences sector. I'm Don Alexander, founder of GeneCoda, a boutique executive search firm dedicated to connecting visionary leaders with life science companies, shaping the future of therapeutics and scientific tools. On each episode, we'll dive into candid conversations with industry pioneers, uncovering strategies, insights, and real world experiences that drive success in our industry. If you're interested in presenting as a guest of a future episode, please scan the barcode image at the top of my video frame and follow the instructions. Now I'm pleased to be joined by Dave Gallup, the CEO of EMPIRI. Welcome, Dave.
Oh, hi, Don. Very nice to meet you. Not meet you, join you. Thank you for the invitation. We've known each other a long time,
Absolutely.
many years, many years.
Many years we've known each other. We appreciate having you on. Dave is co-founder and CEO of EMPIRI, an oncology platform company commercializing eSlice, a 3D ex vivo assay that measures drug response on human liver tumor tissue while preserving the native architecture and tumor environment for more than 30 days. EMPIRI works with pharma and biotech partners to bridge the translational gap between animal models and clinical trials, and with academic centers, including Houston Methodist Research Center and MD Anderson, on prospective studies of immunotherapy and targeted therapy response. The company is supported by an NIH, R01, an NSF SBIR, and a CPRIT product development seed grant, and was selected into the 2025 BioTools Innovator cohort. Before EMPIRI, Dave spent more than 2 decades building biological analysis businesses. He co-founded MLab P&K and grew it into one of the largest commercial indoor air quality laboratories in North America, leading it through acquisitions by Test America and then Eurofins, and being recognized as Business of the Year 4 times. He's a graduate of Caltech and Stanford and named inventor on 9 patents across 4 industries. So again, Dave, welcome. Um, let's jump into your career journey and leadership background. First of all, what led you into the life sciences and eventually to co-founding EMPIRI?
Oh, I see. Well, the primary driver is actually my wife, Kyusan Yoon. She's a brilliant scientist. Her father actually one day said, "Oh, there's a lump in my mouth." And then it turns out that was diagnosed as metastatic lung cancer. And they didn't even try to treat him with anything. They went straight to palliative care. And 3 months later, he had passed away. And that was kind of a transformative event for her. And she's dedicated the last 25 years to kind of studying cancer care and trying to find a way so that patients at least have some care available. So right now it's kind of remarkable that 25, 30 years later, we have hundreds of FDA approved drugs and no effective way to deploy them to patients. And so I had been running, as you mentioned, biology, environmental microbiology labs, 18 locations across the country. And I was finishing up my career there. And she said, I have this great idea that addresses that need of helping patients directly with a very precise care tailored for them from among the drugs available. And so we started EMPIRI to try to address that unmet need in the market.
That's great, Dave. Yeah, so many of the entrepreneurs like yourself that we've spoken with on this show are really empowered by personal stories, in terms of their own journey, their own lives. It drives them to do these things. So, thank you for sharing that. What is the unmet need or moment of conviction, rather, that made eSlice worth building a company around?
Right. So I think the unmet need, which really is really so many patients go in for standard of care treatment, and that standard of care isn't tailored to the individual patient. It's really tailored. It might be, oh, I have triple negative breast cancer. Oh, I have pancreatic ductal adenocarcinoma. Oh, I have these different things. And that standard of care may say, pick one of these choices. Or it may pick a treatment that's 40% effective or 50% effective. And so then you're spending time, money, and delaying treatment, just trying different things with the patient. And so there are many cases where we can, with eSlice, we can take their biopsy that's part of standard of care. They go in, they get a biopsy taken. We can then treat that, we can test that in the lab, and typically in 8 to 12 days, tell that patient what their particular tumor will or won't respond to. And so that has tremendous value to kind of the patient, to the oncologist, and to the payers, and sometimes with the drug companies. As we have been running EMPIRI, we're also addressing a second large unmet need, which is in the drug discovery and development pipeline. So we can help many aspects of that pipeline, but the most obvious one that we're addressing right now is the gap between animal models and in human clinical trials. Because we provide a platform in which you can test the drugs in the lab in human tissue with what's called an intact tumor microenvironment, which we can discuss. It's important. Anyway, so we are as close as you can get to in-human testing, where the risk is lower, the cost is lower, and then direct either patient selection or adjust dosing or pick the indication to reduce risk, reduce cost, and shorten then the clinical trial cycle before. And that's when the real money starts to impact, is when you go to end, because inpatient clinical studies can be 150 grand a patient, for example. I mean, there's a huge range around that, but it's very expensive per patient.
That's great. So really you're using it at that kind of translational segment, preclinical trials. It sounds like with respect to the biopharma industry specifically, and then it's also got clinical utility in helping patients understand their more precise tumor type.
Yeah, so we will help them select the correct or the optimal treatment for the patient as a cancer diagnostic, and then we help the drug development process.
Excellent, excellent. Can you maybe elaborate on eSlice in practical terms? What happens to the patient sample? What kind of response data is the company able to generate?
Sure. So what happens is the patient goes into the clinic, and depending on their cancer type, but they may get either a biopsy, a needle core biopsy taken, or a surgical sample taken. If they send us a portion of that tissue, we then in the lab can precisely cut it into very thin slices. And then we can expose those thin slices of their tumor to different drugs. And then we can measure the response of their tumor to those drugs, the choices that the oncologist is considering over time. And that time frame is typically, depending on how the drug's mechanism of action is the term, what it's doing, typically between 5 and 12 days, so a clinically actionable time frame. We can then say this tumor is responding to these drugs or this drug, and it's not responding to these drugs. And we can provide that to help guide the oncologist in selecting the treatment that's best for that patient. And the key, many people, it's kind of an obvious thing to try to predict personalized care, to try to predict treatment response. People have been trying to do this through a variety of ways for a long time. What's different about eSlice is we maintain what's called the tumor microenvironment. So cancer, the world's been trying to solve cancer for a long, long time. This kind of, I think it's important. Is it okay if I go on with this?
Please.
Well, so the cancer cells respond to their environment. So even if you have a genetic profile, that environment can change how the cancer responds. So for example, if I were to take you and drop you in downtown New York, and I were to take you and drop you in the Sahara Desert, it's the same Don Alexander, the same genes, the same life experiences, the same person, but how you react in that environment is dramatically different because of what's happening around you. And it's the same with the cancer cells. You can genetically profile them and find out what mutations they do or don't have, but how they respond is also influenced often strongly by what's happening around them. So if you look purely at the genetic profile, or you take that cell out and deal with it in isolation, that prediction is often going to be flawed. Whereas with eSlice, we're taking the intact tumor, intact with the native stroma, with the native immune cells, the cell signaling, everything that's happening around those cancer cells, and we expose it to drugs in that environment. And so our prediction is much more accurate than the other commonly available methods.
That's really helpful, Dave. The environment is critical, as you say. So, in understanding that EMPIRI emphasizes live tumor tissue and the preservation of the tumor microenvironment, why are those features, do you feel like, so important in oncology drug response testing?
Well, so their importance is, I guess there's a couple of things. One is the cancer cells are strongly influenced by what's happening around them, and some treatments aren't directly impacting the tumor cells or the cancer cells. They may be utilizing native immune cells, for example, or native T cells or other things. And so if you're looking just at cell lines or organoids, they lack that native immune cell population that's in the tumor. And our platform, eSlice, has those intact. And so now we can measure what's the drug actually doing, whereas that's not possible in other platforms.
Got it. Excellent. Excellent. You mentioned earlier, touched on genomic profiling and maybe some of the limitations. How should individuals, executives, people in general think about eSlice relative to, say, genomics, organoids, patient-derived xenografts, other 2 or 3D model organisms?
Oh, I see. Yeah, there's a whole platform of tools, and they all have kind of their place within either patient care or within the drug discovery and development pipeline. So 2D cell lines are cheap, they're quick, and they are very good for early drug discovery to do screening of multiple different drugs, for example. You can then move to organoids, which add complexity. They add a 3D element, and you're now a little closer to the human, but they still miss the immune cells. They don't have a native tumor microenvironment, so they have their place, but they still have a gap. You can then move to what's called patient-derived xenografts. This is where they take the human tumor and they put it in a mouse, and then they grow out that tumor in the mouse. And so now you can have a circulatory system, you have an immune system that has more of the complexity of kind of a real tumor, but it's still not human tissue. And so you're closer, but animal models have many flaws, as is being recognized by the industry now. And so then you have in clinical trials, and there's that gap between the animal models and the human trials. That's where eSlice drops in very nicely. eSlice has other utilities, but that's where we're helping pharmaceutical companies right now. And then genomics helps kind of along much of this platform because they can understand what's happening in the animal. They can help understand what's happening within the eSlice. They can help understand what's in the patient. But genomic testing or molecular profiling has limitations. So it's a huge win in situations where there's one molecular feature that largely determines the right drug. So this is the case with EGFR in lung cancer, BRAF in melanoma. In those cases, sequencing has changed how patients get treated, and this is great. Unfortunately, usually the cancer is more complex, and for most tumors, understanding the microenvironment or resistance mechanisms or immune dynamics means the genomic profiling is helpful, but it's not predictive, it's more probabilistic. And so that's another spot where eSlice could fit in. But all those tools have their place. They all are useful, they're helpful. We just fill in a gap that's missing with those tools.
Got it. Excellent. Let's talk a little bit about drug development validation, commercial strategy for the company as well. What evidence do pharma partners or clinicians need before they change how they evaluate a therapy or make treatment decisions?
Oh, yeah. So it's very situation dependent. So currently, we, for the pharmaceutical companies, we have contracts with, I don't know, over 15 different pharmaceutical companies at the moment, trying to fit that gap between animal models and in human studies. And those meetings, those sales calls, if you will, start with talking to their scientific team to evaluate the platform. And we show them we have single cell sequencing data, we have histology data, H&E, we have all sorts of data to show them that our platform works very well and is predictive. And so then they say, great, here's our drug. We're interested in this cancer type. We want to do this type of test. And then we go help them out. For oncologists, the data they need, I think, is pretty broad. So if we are trying to change standard of care in a cancer that's readily curable and solvable, I don't think much data is going to change their opinion. If it's a case where we're if it's a case there's no standard of care, I think that bar is lower. If it's a case where standard of care says, hey, pick between A, B, and C, and there's not a clear winner, they're all 50% effective, then those are discussions we're having now to try to run smaller pilot studies. I think that because we're guiding care and we're sticking within standard of care in those situations, we're not exposing the patient to more risk. We should be decreasing risk and increasing their odds of success. So I think the threshold is lower. But right now, we've just had a publication in the fall with MD Anderson. We had a poster with BioNTech in the fall. We have some early data with MD Anderson with our R01 showing our predictive accuracy with triple negative breast cancer. We were 9 for 10 in the interim study. We predicted treatment 9 out of 10 times correctly. So I think that data helps oncologists kind of approve it, but it's situation dependent is the short answer.
Gotcha. Gotcha. Excellent. And for drug developers, where can functional response data most directly reduce risk? We touched on this earlier, but their earlier candidate selection, dose optimization, combination strategy, or trial enrichment.
So where if we say, hey, the market's deciding what they want right now, the market is saying they want in that gap between animal models and their clinical studies. I think once they get comfort with it, because it's still early, I think they all see the huge value in helping guide clinical trials. So clinical trials, and that's, I mean, as I said earlier, that's where the expense comes. And if we could, for example, say, hey, you give us a tumor that you're taking, a piece of it, we'll tell you whether that patient will or won't respond to your drug. Now we can help guide patient selection for that trial. And so now they can get a cohort of patients that responded favorably to that drug. So it's more likely to get FDA approved. The concern with that is that then we would be called what's called a companion diagnostic, which means if you're going to prescribe the drug, you have to run eSlice prior to it, because that's how the clinical trial was run. So we like that, but pharmaceutical companies don't like that. And so but it's kind of an obvious example of the value that we can add there. We can help them pick which indication is most useful. We can help them understand why patients did or didn't respond to their drug in the trial to kind of guide further refinement. And once we get into the clinical setting, we can add tremendous value. And there's value to be had, but it's painful to them of also failing early. We had one pharma company where we said that drug's not going to work. But they have all this animal data saying it is going to work, and they're ready to go to the expensive part. and they went ahead and it didn't work. But the problem is we're early, so they're going to need much more data to convince them to pull the plug, on a big program like that. So that's up to us to generate that data and generate the confidence that the platform works that they need.
Absolutely. Yeah, expanding, I can see that in expanding indications, but also the pain of having to pull programs, although fiscally that's the right thing to do. But
Yeah,
um, what are
fail
the,
early.
yeah, uh, never easy. What are the most challenging parts of scaling a live tissue assay platform? Sample logistics, reproducibility, turnaround time, something else?
So I think there's two, because we're really talking two different markets. One's the farm helping drug development, the other is the cancer diagnostic.
Yeah.
And the constraints are different. The constraint with the helping the pharmaceutical companies is actually getting the tumor tissue. So typically they say, we're interested in PDAC or renal cell carcinoma, and then we need to go get the tissue, and that's the bottleneck. If it's a cancer diagnostic, then they're sending us the tissue. And so then the focus is more on scaling. So, and that's what we're getting help with the NSF is on automating the assay so that when we're successful and we're trying to do thousands of these, we can scale it up and control with all the benefits of automation. Cost, speed, quality, automation is great.
Yep, absolutely. Absolutely. No, thanks for that. Let's talk a little bit about precision oncology in general. So precision oncology, Dave, has long leaned heavily on molecular profiling. Where has that worked well, and where are the limitations becoming clearer?
Mm-hmm. So um, molecular profiling works best when there's a kind of a single molecular feature that will select the right drug. And there are some narrow indications where that is great and has worked well. Um, uh, but unfortunately, most of the time, the cancer is more complex. And so it becomes more probabilistic in nature rather than deterministic. And so then eSlice does not replace, but it can supplement that that genetic information. So the genetic information can narrow down the number of drug choices, and then we can test them in eSlice. Because one of the limitations of eSlice is, unlike cell lines, we can't test thousands of different drugs or all 100 or 300 FDA approved drugs. We're going to be better off if you say test these six or
Right, right.
so genetic
Absolutely.
profiling can reduce that list for us.
That's yeah, absolutely. So yeah, really, they work. It's a symbiotic relationship, really, almost between the two.
Yeah, I don't
Um,
think
yeah,
eSlice, I don't think is competing with the current technology. I think that they
yeah,
all have their utility and there's just a gap that we drop into very nicely.
yeah, absolutely, absolutely. So if Empiri is successful, what changes for patients, oncologists, and therapeutic developers do you think?
Oh,
Looking out a few years, maybe.
yeah. Well, we're ready to go.
Yeah,
so, so I think the patients will get the treatment that they're going to respond to the best, and they will avoid treatments that they will not have benefit from. Just one example because it's relevant. I mentioned earlier we were correct in 9 out of 10 in an interim data. So this is with MD Anderson and Houston Methodist. They that treatment is for TNBC, where the standard of care is patients get what's called a keynote 522 regime. So there's 25,000 patients a year that get TNBC. Standard of care is this regime. That regime, roughly speaking, is $150,000, right? From those 25,000 patients, roughly 20,000 will not respond favorably to that drug. 20,000 times 150 grand is $3 billion being spent on care that's not going to help those patients, and in some cases harm the patients. And so we can avoid that. So that's good for the patient. If we stay within standard of care, the oncologist gets to pick the right drug, and they're not saying, hey, go off label, do something odd. We're not exposing to the risk. They just get the right treatment for their patients. It's helpful for them. It can help payers, as mentioned before, and it can help pharmaceutical companies whose drug is going to help that particular patient. I mean, it's a win kind of for everybody. And it's just up to us to add to the data we have to get people comfortable with its use.
Absolutely. That's great. So on our podcast, we have a lot of innovators and individuals who've founded companies in a broader sense. Just given your experience really across the board, formerly and with EMPIRI currently, what advice would you give founders working to bring a scientifically complex platform into clinical and, in this case, biopharma use?
I would say get expert advice. it's it's a complicated environment. So try to, and there's all sorts of people that are willing to be helpful, provide advice. The talk, be sure that you really understand your target client and their unmet need. So go out and talk to the actual users and be sure that your unmet need is real and that they're willing, and it's large enough that they're willing to pay for it.
Mm-hmm.
And then try to make your decisions data-based, use the best tools available. With AI, it's incredible, right? You have to use it intelligently, thoughtfully, cautiously, but the tools that are coming online to help with things are stunning. And then try to minimize risk. And the risk is going to depend on the company. It might be revenue, it might be market strategy, it might be, I mean, it's all sorts of things. But kind of looking at all this, our mission is, EMPIRI's long-term mission is to change cancer care directly, be a cancer diagnostic, select patient, personalized patient, drug selection. But early on, when we were trying to raise money for this, the investors frankly said, cut that out. They said, that is long, is expensive, it's risky. FDA is out of your control. At that time, they're buried with COVID. They said, you're not going to raise money doing that. They said, switch to be a CRO, help drug development companies. And so we said, okay. And here we are later, and we have contracts with 15, 18 different companies. We're cash flow positive, and that gives us time to continue our path to our goal without being in a cash crisis and a crunch and everything else. So that's an example of where the advisors just, gave us very easy advice. So easy is the wrong word, but very good advice. They just, what the advice was, just cut it out. You're not doing the right thing.
Well, you've done an admirable job of listening to the market, is what that sounds like to me. I guess final question is, how can listeners connect with you and learn more about EMPIRI?
Oh, easy. So our website has EMPIRI Cancer. Our company name's EMPIRI, but the website is EMPIRI Cancer, EMPIRI cancer.com. EMPIRI is short for empirical. So they can either email info@empiricancer.com, or our website has the normal form. You put in your email address, your question, or actually our website also has a live link to schedule a meeting if you want to do that directly. So any of those methods work.
That's great. Well, that wraps up, Dave. Thank you. That wraps up this episode of Executive Insights for Life Sciences Innovators. I appreciate you tuning in and hope that you found today's conversation valuable. If you enjoyed the discussion, please be sure to leave a review and share it with your network. Please also mark your calendars for our next session with David Pass of Alteryx Bio on June 15th at 1 o'clock Eastern Time. Thanks for listening, and I'll see you soon.